kind of just the general life cycle or viral reproductive cycle. Of course, you're going to start out with a virus, simple little virus, that's ready to infect and cause issues. Of course, the virus is going to have a couple of important parts. Inside of it, we see some type of genetic material. Remember, the genetic material could be possibly DNA or it could be RNA. We're not 100% sure on each case. It's different for each virus. And then there's a capsid on the outside. And the capsid, of course, is made of protein. Those are your two major structures, the two things we should notice the most. Now, of course, here is a picture of the cell that it's going to invade. So I think it would be appropriate that we call this cell a host cell. It's going to be housing the virus. Now, one thing you'll notice is that the virus is very, very easily going to be brought into the cell. Now, that's going to make sense because where do you think this virus particle has come from? Another cell for sure, another host cell that is the exact same type. So I want us to really appreciate and understand that the proteins that would be on the outside of a host cell, because the host cell is responsible for making those viral particles, the proteins on a new virus should match the proteins on the host cell. Good? So we could agree that there's got to be some kind of protein complement to allow the virus to come inside. Now, once it does, and the virus is now brought in, you'll notice that everything is seemingly broken up. All your little protein capsid pieces are degraded into protein chunks, and your viral genetic material is placed right there. So this would be your viral genetic material. Now, there's a couple of different things that can happen. Somehow, that viral genetic material is likely to be placed into the cell's original genetic material. So I could write here that there is an incorporation, or we're going to incorporate viral genetic material into host DNA. So the host DNA is reprogrammed because genetic material from the virus is placed inside of it. And also, that viral genetic material will be transcribed. And what do we create after we do transcription? Okay, mRNA. So there's mRNA, you see right there. And what do you do after you have mRNA? What do you do to that? Translation. And what do you make after translation? So there's proteins. Now we know that at the start of this process, the host cell had some elaborate and its own personal function, and now that function has been turned off, and this host cell now has a purpose of reproducing viral particles. So I'm going to write that the sole purpose is now virus production. In fact, what you see here in this step is the coming together of DNA pieces that have been synthesized and the proteins that have been synthesized, and there's like a self-assembly system that occurs. And that assembly of the new viral particles that of course are released, and there's your new virus particles that of course will attack a different cell altogether. Same type of cell, just a different one. Yes, ma'am?
So is the first host cell like the hardest for a virus to interact with because it's not like accustomed to whatever protein it has to bind to?
Not really, because like if you think about a case of like HIV, like the HIV, you know, if an individual contracts HIV, it's pretty obvious that it's not coming from thin air. They're contracting it from another individual. The other individual that gave them HIV, it was their T cells that had been reprogrammed, and their T cells were producing the HIV particles. It's for sure certainly the HIV particles that are now in the new individual. So it's not about your proteins, it's about the typical proteins on the outside of that type of cell. So HIV particles just seemingly are very, very adapt and able to bind to helper T cell proteins. So it's like this perfect complementary fit. So even if you jump individual to individual, the getting in is the same. Cool? Okay. So I think that's a really, really good picture. And the next thing I want to go over is I want to go over two types of cell stages. And the first stage is going to require drawing, and so is the second stage. So for this, I'm already apologetic. Very end of this lytic cycle, you've got the death of the host cell. So we could write up top lytic cycle, and that this will result in host cell death. So the host cell is going to die for sure. A virus that produces by this type of cycle, this type of virus that uses the lytic cycle is called a virulent phage. Virulent. The virulent phage is, resulting in the death of the host cell. Okay. So in this drawing, you can obviously, right here, there would be like an attachment phase. And that's attachment of the virus to the host cell. Obviously, there's going to be, and you can see it kind of in this drawing and in this drawing, you've got the releasing of viral genetic material. And it's at this point that the cell loses its native or natural function. The cell function is lost. And its new function becomes to just be making new phages, making new viral particles. So with this new function, this host cell is now a center of viral production. And so you have, over the course of this time, the assembly of viral particles. So there's that assembly of the viral particles. And at the very end, what happens to the host cell? Host cell kind of explodes, right? Yeah, host cell lyses. Of course, this would be releasing viral particles. Viral particles go out because the host cell is dead. Now, this is a unique type of strategy because, of course, it results in host cell death, but the death of the host cell is also the mechanism of releasing the viral particles. Problem is that the virus has just self-destructed its manufacturing center. But I guess the positive is that now there's more viral particles that can attach to more host cells and then cause the death of those other host cells. So they will for sure take over the system. Yes, sir.
How, when you have a virus like a cold or something,
Mhm.
how do you ever get rid of it? Because that seems like an ongoing process.
What we see in this drawing is kind of a combination effect of the lytic cycle and then a different type of cycle. You could recognize over here that you've got something that's called, or that is in the lytic cycle, because you can see that the host cell, of course, is popping at the end. The cell's dead. And then over here we have a different type of cycle. This would be the lysogenic cycle. And you'll notice that in the lysogenic cycle, your host cell at the end is still alive. It's like the DNA is incorporated over here, and then over here you're just all about making new viral particles and releasing them. So I want us to understand that you can have both of these going on, even in the same virus. Some viruses have the ability to if you will, go around and do both of these types. If you have a virus that can do both types of the cycle, we would say that that's called a temperate phage. A temperate phage can convert between the lytic and lysogenic cycles. Cool. The difference between lytic and lysogenic.